Without further ado, we're going to go ahead and get started with our speaker. I want to ask you to unmute. Okay. Dr. Wilhelm, like I said, again, we welcome you and all your accomplishments and your work, and we look forward to hearing your talk. Dr. Wilhelm.
Thank you so much, Dr. Mendenhall, for inviting me to be here and for that wonderful welcome. It's a joy to be here. For those of you who don't know, Morehouse was one of my intellectual hearts. So I did my grad school and postdoc at Emory University, and my first job out of grad school and when I was a postdoc was to teach psychology at Morehouse. And it was one of the best experiences of my life. I really treasured the mentorship I got from my colleagues in the psychology department, particularly Dr. Tina Chang and Dr. Jackson. So I'm glad to be back. I wish I was on campus and able to talk to you, but it's nice just to be able to spend this time with Zoom with y'all. I just want to tell you a little bit about what I've been doing since I've been at the College of Charleston. I joined the faculty here in 2012 and am now an associate professor. And during my time there, I've mentored a bunch of students, and I wanted to talk to you a little bit about the work that they've accomplished and that I've accomplished, and to share a little bit of the history that I have and some of the challenges we've met along the way. So with that, I'm going to share my screen. And I also want to tell you, if you have any questions, feel free to stop me or to put them in the chat. I'm always happy to answer questions along the way too. So today I want to talk a little bit about the novel therapeutics that I have worked with my students to develop. I'm interested in peripheral nerve regeneration, and we'll talk a little bit about that, and the different therapeutic options that can be used to help people who suffer from peripheral nerve injury. So the first thing we need to talk about is a little bit of a neuroscience review. Some of y'all may have already know this, and some of this may be new, but generally when somebody hears neuroscience, they think about the brain, and that's totally fine. The brain is super important, but the brain is just a small portion of our nervous system. So we have the central nervous system, which encapsulates the brain and also the spinal cord, but we have this rich system of nerves that come off the brain and the spinal cord that allow us to talk, to walk, to feel. I know we're not getting to feel a lot these days. We're not getting able to give people hugs and to do a lot of touching, but the peripheral nervous system is what allows us to do that. And these are all the nerves that come off the brain and the spinal cord. Unlike the brain and the spinal cord, the peripheral nervous system is not covered in a nice bony structure. So you can see here as a cutaway of the spinal cord, and we can see that we have here, you can see that there's a motor neuron, which is one of the neurons that is in our spinal cord that has an axon that goes out to connect with a muscle. And if we look down here, you can see that motor neuron would be safely housed inside of that, inside the spine, but the axon that forms that nerve is going to go out into the periphery. And the same with the sensory information that's coming in from your skin and from your muscles that help us to feel temperature and pain and touch and muscle stretches. Those are going to be coming in, and then they get protected once they get to here, but outside they're not protected. So unfortunately, that means that about 100,000 Americans every year suffer from traumatic injury to their peripheral nervous system. This is a big deal. So 100,000 Americans are suffering from traumatic injury. And unfortunately for most of them, they don't actually fully recover. Less than 10% of people fully recover from a peripheral nervous system injury, a traumatic one. And actually, I would like to talk about how the statistics are maybe a little bit underrepresented. I can guarantee that everybody on this call has sustained a peripheral nervous system injury at some point. So if you've ever gotten a deep paper cut, if you ever cut yourself, congratulations, you have had a peripheral nerve injury. For the most part, the nervous system is able to fix itself, and you're not left with very big trauma. Your skin, for a small paper cut, your skin grows back, and you don't really notice a detriment. I had a more serious injury. So this finger right here, I cut the tip off of several years ago. We were able to sew it back on, but I've been left with some deficits. My fingertip just doesn't feel right. It feels kind of like when it's really cold outside and you come inside, and that kind of burning numbness, that's what this fingertip feels like all the time. So one of the things I'm interested in is, what do we do to get the nervous system to regenerate better so that people aren't left with those differences? The nerve that I study is the sciatic nerve, and it starts Its motor neurons, the cell bodies, are here in the middle of the lumbar area of the spinal cord. So kind of right there along the middle of your back. And those axons come out. The motor axons come out and they go down the leg, down into the thigh, to the calf, into the foot. And the motor axons help with muscle contraction and for foot dorsiflexion, so for walking and stepping. We also have sensory neurons coming out of that, and that innervates all of these areas and gives us all those sensations and the ability to stretch our muscles, to feel the floor when we step on the floor. It is one of the largest nerves in your body, and it's also one of the ones, unfortunately, that gets damaged fairly often. So people deal with sciatica, which is irritation of the sciatic nerve. They have cuts, traumatic injuries. And unfortunately, there's not a lot of good options. Like what I had encountered, the only option really is to have surgery. So most people who have these peripheral nerve injuries end up going to a surgeon, and the surgeon will do the best they can to repair the nerve. So if you're a little squeamish, look away. But I wanted to show you what a nerve might look like. So here's one of these nerves, and you can see that this person actually is pretty lucky. They had a fairly even cut along that nerve, and the surgeon was able to sew it back together. You can see some sutures right here, and they were able to repair that nerve. This does not mean that it's going to be fixed and back to normal, and we'll talk about why, but you are able to kind of put them back together most of the time, unless you sustain a really big ripping injury, and sometimes that happens, especially when people are in car accidents, or you have a big cut or something that's not just a knife. You may be left in a situation where you have a bit of a gap, and it's not possible for those nerves just to be sewn back together directly. And in this case, a surgeon might have to harvest nerve from another part of your body. They may take a smaller nerve and put it in. They may take a cadaver nerve, and they might put a piece of nerve from a cadaver in there. Or, which Dr. Mendenhall might be interested in, they might use a biotechnology. So they might use some type of biomaterial that they can then place between that gap to allow the nerve to be redone. But that's about it. That's the only treatment that's available right now. So people go to the surgeon, they get sewn back up, and they were turned loose, because when you cut those axons, those motor axons and sensory axons, you now have a break in those axons. They can't send messages down to their target muscles or from the skin back to the cell bodies that have sensation. So people are left with a lot of deficits. So we have here a picture of a motor neuron. So this was one of our motor neurons that's going to attach to our muscle. And normally it has an axon that goes down and it has a connection here to the muscle. And in an injury, like the one we just saw, that gets cut. So the axon gets cut. And then the part that's attached to the cell body up here, that is going to remain alive, and it's going to remain able to have cellular function. And the cells around it, these are called Schwann cells, are going to be able to stay myelinated. So they're wrapping around this axon really tight and allowing the conductances to be so that we can have action potentials. But the part that is distal to the cut, the part that used to be attached to the muscle, it actually degenerates. It goes through Wallerian degeneration, and the axon blebs, it comes apart. Those Schwann cells come off and they become reactive. We get infiltration from macrophages, and we have an immune reaction occurring. We also have these cells starting to release growth factors. So these are going to be different proteins that are going to encourage, luckily, this axon that's still alive to start growing back. And eventually we end up in a situation where the axon grows back, the Schwann cells remyelinate, and we can get back to a situation where we have that target muscle reinnervated, so we can get it back to normal. Unfortunately, this doesn't happen all the time. Not all axons are able to get back. It depends on how far that injury is from the cell body or from the target organ. So something like a paper cut at the tip of my finger, or cutting my finger off, that actually didn't take that long to regenerate, because even though it's really far away from the cell body, which is up here around my neck, it didn't have very long to the target organ. So that only took maybe a few weeks. If somebody has a larger injury, like in their brachial plexus, and that's going to be the main nerves that go all the way out to your fingers, that could take years. An injury in your pelvis or in your hip that would be to your sciatic nerve, that could take years to have this process happen. And unfortunately, those growth factors, they don't stick around for years. This process of wall regeneration and the growth factors coming out, they usually only last for about 3, maybe 4 months without any intervention. So this process happens. It doesn't keep happening for very long. So a lot of people are left with situations where they don't actually reach this state over here, which is full reinnervation. They may be stuck in this state where they have a little bit less. So some people who are lucky, they can have reinnervation happen, but we still have an extra challenge. When those doctors, when we looked at that surgical repair, that nerve is really big. And that nerve, what we're looking at the outside, is a bundle of axons. It's just the fatty casing. And the axons themselves are microscopic. We can't really see them without a high-powered microscope. There is no way that a surgeon can put them back together and have those axons get into the right tunnels that's going to lead it from where it started to where it's supposed to go. So occasionally an axon might do the right thing, but often it ends up with having a situation where misdirection occurs. So I'll show you a little bit of that. So this is a normal nerve. It's going to be a bundle of axons. These are all going to go to skeletal muscle. So these are motor axons, but remember our nerve also contains sensory. So here are our Schwann cell covered myelinated axons going to a target skeletal muscle. After the injury, we're going to see degeneration of this side. But those tubes, those tunnels, the actual white fatty stuff that we saw, that stays there. So you can connect that back, but the axons have to regenerate. And you can see this axon from here, it makes a little bit of a turn. And instead of heading back down the tunnel that it's supposed to go through, it takes a turn that goes down this tunnel. This tunnel may lead to the same muscle that it needed to go to before, or it could go to a different muscle. It could go to an opposing muscle. So we remember some of our muscles need to contract and others need to relax. If this one goes to the wrong muscle, you may get contraction when you're supposed to get relaxation. It could also go to skin. That would be bad to have a motor neuron going to a sensory organ, or a sensory organ, a sensory neuron going to a muscle. Ouch. So then you think you're going to be, your muscle contracts and it sends a negative sensory information, or it might send pain. So this misdirection ends up causing a lot of the functional deficits that people are left with, because you don't end up having the same, even if the regeneration happens, it doesn't necessarily happen accurately. So this leaves patients with a lot of issues. You may have weakness, especially if we're looking at sciatic nerve. People might have weakness. They may not have a clear gait. So we use this nerve for our walking. So you may shuffle, people may waddle, they may do something called foot drop, where they can't actually pick their foot up and carry it across. And really, in a lot of cases, if they end up having the sensory neurons not grow back together, they may not be able to sense that their foot has touched the floor. And that's incredibly important for walking. So you can get, say your muscles were reinnervated properly. If your sensory system doesn't, then you put your foot down, but you don't know you put your foot down. So how do you know you can support your weight? So functional recovery is a really important thing. And right now, there's not much that a lot of the doctors can do. They send people to the surgeon, and then they say, good luck. So a lot of times when this nerve has been damaged, you'll get muscle wasting, because I showed you that it's no longer connected to the muscle anymore. And the muscle, it really needs that connection. So you'll get muscles to start wasting away. You'll get them to become smaller. So doctors would say, hey, why don't you go to a physical therapist? And a physical therapist sounds like a great idea. They can work to try to build that muscle up. And a lot of physical therapists were giving great exercises. But we didn't really know, what is this physical therapy actually doing for these neurons that are being damaged. So I worked with a team at Emory University. I worked in Art English's lab with some people who were interested in physical therapy, and also an exercise physiologist. And they were interested in how treadmill exercise has been used as a therapeutic for a lot of patients who have had sciatic nerve injuries. Remember, we also have the sciatic nerve comes down right along here. So it makes sense that a lot of these patients are put on treadmills. So we don't use humans in our research. That's not really considered ethical. Not many people want me to go in and cut their nerves. So we use a mouse model in our lab, and I'll tell you a little bit about some different experiments that we do. So we use both male and female mice. These were not pictures that I took. These are representative pictures. because iCook does not like us taking pictures of animals. But you can see that this is an animal that has undergone a surgery to expose the sciatic nerve. So this is what it looks like. This is one of the branches, probably the common fibular branch of the sciatic nerve. As you can see the muscles being retracted. Here you can see a little bit better. The nerve has been cut, and then they were able to repair it. So you can't really see it because it's white on white, but they are able to repair it. We do the same thing in our lab. We anesthetize the animal, expose the nerve, cut it, and then we repair it using something called fibrin glue that basically glues the nerve back together. Then to test to see what exercise does, to see how exercise might be effective, we use two different training paradigms that were used by physical therapists. One is going to be a continuous paradigm, is what I'm going to call it, and it's a slow walking. It's 10 meters per minute for 60 minutes a day for 5 days a week for 2 weeks. 10 meters a minute is super slow for a mouse. Their top speed is about 35 meters per minute. So 10 meters per minute would be like us slogging along very slowly. This is not taxing for them. So they can, 2 days after their surgery, we start them on the treadmill, and they are usually running the entire 60 minutes at this speed. The other paradigm that we use is an interval paradigm. These animals run twice as fast. They're at 20 meters per minute, and they run in intervals. They're 2 minutes in duration at 20 meters per minute, and they have a 5-minute rest, and they do 4 repetitions of this. And these are chosen because these were two different paradigms that were used commonly by physical therapists who are treating sciatic nerve injuries. And what we found, and these are my colleagues, Dr. Art English at Emory University and Dr. Jill Ward, who is also at Emory University, we found that exercise in the form of treadmill training will cause axons to grow longer than if an animal did not have exercise. So in this case, the mouse that we used had a special protein that allowed it to some of the axons to express a yellow fluorescent protein. So we were able to have this animal. We could cut the nerve, and you can see it over here. We cut it, and then the piece of nerve that we put in to repair it came from an animal that did not have that yellow protein. So we could basically create a black or blank background in which any regenerating axons growing past this cut mark right here would be yellow. So, and here you can see, here's my control animal. This was the proximal segment that was still attached to those motor neuron cell bodies. Here's the line where the cut happened, and then here's the graft, and you can see that axons do grow through it. So these axons were growing. You can see the little growth cones down here at the end. So in this control animal, we see about 1,000 microns of growth. When we exercised the animal, it went up to about 3,000, 2,500 microns. So that's a huge increase. So we saw a very large increase in the way that exercise was able to get axons to regenerate. We did not look at specificity in this case. I'm not going to talk about specificity anymore, but we were able to get the axons to grow longer. We then had a situation, and several of you, if you're students who work in research labs, you may have, when you joined, been asked to try to redo some of the experiments or do some control experiments just to kind of learn the techniques and to learn about what you're doing. So we had some students that we asked to join the lab, and we gave them some experiments where we asked them to treadmill train the animals, either using that interval paradigm or the continuous paradigm, and they could not replicate that data. They kept coming and saying, this isn't working, this isn't working. And we showed that's a pretty dramatic difference that we showed in this right here. So we did a little deeper investigation, and we discovered that one of the students had always pulled male animals, and the other student had always pulled female animals. And before, we had combined them together. And now we can appreciate, based on their research, that male and female animals actually act differently. They have different kinds of regeneration. And this was done by Kylene Wood and Kevin Liu. They are both physicians now. They graduated several years ago, but this was their work that they did. And what they discovered is that untrained animals, male and female, they both have axons that grow to about 1,000 microns. But when you give that continuous training, that slow training to males, they have this remarkable increase to about 3,000 microns. Whereas females, it doesn't work at all. And we didn't appreciate the before because we had not separated the sexes. So this is one of the trial and errors that we learned along the way, is that the sex of the participant does matter. And this was actually the very first time that anybody had shown this. And this was two undergrads that I worked with. So this was somebody that, they were still undergrads that made this discovery. And it was the very first time that anybody had ever shown a sex difference in this way and in these cells that are not sexually dimorphic. These are not supposed to be different between sexes. This was a remarkable finding, and these were undergrads who did it. So we then looked to see, well, what happens with interval training? And we found that, in fact, the interval training worked in females, but not in males. And so we now found this strange sex difference. And we started wondering, what's going on? And I think of the answer. I'm not going to share it with you quite yet because I want to show you a few other things first, but I'll come back to it. So we've shown elongation has worked, and that exercise works for elongation. So it actually enhances the length of these regenerating axons. But that means a little bit, but not a ton, because I don't know if you noticed in one of these pictures back here that when these axons, when they regenerate, they can actually sprout. So this little guy right here is going into 2 different tubes, whereas before he only went into 1. So showing these axons are longer could mean that we're getting them growing longer, but it could mean that we just have 1 or 2 neurons that are growing a whole bunch of sprouted axons. So the question became, well, how many neurons are participating in this regeneration? So this has been something that I worked on in my lab at the College of Charleston. We have an animal model as well, where we expose the sciatic nerve, we cut it, we repair it with fibrin glue, and then 2 weeks later, after we've exercised the animal or treated it otherwise, we come back and we cut it about 1,500 microns, which is, of course, a little bit longer than those control animals I showed you before. And we put in a retrograde dye. And this dye binds to dextranamine. It's dextranamine conjugated, so it binds to molecular motors in the axons, and it carries that dye back up to the cell bodies. And 3 days after we do this dyeing, we are able to collect the tissue, and we can count the number of dyed cells or labeled cells in both the spinal cord to look at motor neurons and in the sensory neurons in the dorsal root ganglion. This is the first time people have been able to really separate what's been going on in the motor neurons versus the sensory neurons, because before we were just looking at axons and we can't differentiate between those very easily. So this allows us to differentiate. So what we found, and this was done by Melina Costa, Jamie Harrell, and Trisha Copley in my lab, was that exercise increased the number of labeled motor neurons. So this over here is going to be fold change in the number of labeled motor neurons compared to unexercised and untreated. So that would be the dotted line is this 100 percentile. And then anything above it means more. Anything below it means less than untreated. So you can see for both males and females that exercise increased the number of motor neurons participating. And then we asked, well, is a sex difference? And we found out that yes, just like what we had seen before for elongation, continuous training increases the number of motor neurons participating for males, but not females, and increases the number participating for females, maybe a little bit for males, but it's not statistically significant. So I'm going to kind of call that as a no. So we're still seeing this, the sex difference here in participation, which is really interesting. So we have now exercise is able to get more cells to participate, and the cells that do participate, those axons are growing longer. So what about the sensory neurons? Getting the axons of the motor neurons and motor neurons to participate, that'll give us muscle contraction. But we need to be able to walk. And to be able to walk, your foot has to hit the ground and you have to know it. So we need to look at the sensory neurons. So Allison Bruce was a technician in my lab. She was a non-traditional student who came back and worked with me for several years. And she did an experiment to see, does exercise increase the number of sensory neurons participating? And we found, yes, it does for both males and females. And this was the very first demonstration ever that sensory neurons respond to exercise, which seems kind of crazy because we've known for a long time that motor neurons responded to exercise, but this was the first time anybody showed that sensory neurons respond to exercise. And this was done by an undergrad at the college. So this is really great work that she's done. We're in the process of writing this up right now. And then she asked, okay, do sensory neurons, does it matter to the way that the exercise, the paradigm used, is that going to be dependent on sex? And shockingly, it was not. So exercise in any form, continuous for male and female or interval for male and female, they both were the same. There was no sex difference, which I think is good news because it would have been bad if continuous training worked for males for motor neuron, but not for sensory, or the opposite. So it's kind of good news. It works for both for this. So we were kind of relieved for that reason. So, this is going to be a really important finding, or these are really important findings, because typically when people go to a physical therapist, they are treated for their injury. They go and they say, I have sciatica, I have a sciatic nerve injury. And the therapist says, here are the training, here are the things that you need to do. But this is saying, hey, it matters what the sex of your patient is. So a man and a woman are going to need different therapeutic treatments. So we spent a lot of time trying to retrain the physical therapists and trying to have conversations with them to look at the outcome. Of course, this has all been done in mice, but we are starting to see that this is actually probably true also in humans. So we've been spending a little time that way to work with that. So that was exciting. So this is a potential novel therapeutic. However, there was some bad news, because there always is. We have a real world problem with our research question. So while exercise is fantastic, most patients can't get up and just start jogging within 2 days of having a major traumatic injury. So this exercise is fantastic, but we know that delaying the exercise, this has been some stuff that Art English has shown in his lab, if you delay the exercise, you're not going to get that same enhancement. So the question that we've come up with is basically, if we learn what mechanisms underlie the effects of exercise on axon regeneration and participation, and can we use those mechanisms to design specific treatments that patients can use in lieu of exercise? So I like to sum it up as, I want to put exercise into a pill. So can we do that? How can we do that? So the first thing I'm going to go back to is that finding that we found, that Kevin Liu found, and Kylene Wood, that we have this sex difference between how the males and the females are responding to exercise. So the kind of obvious question was this based on sex steroid hormones? So we took some blood from these animals and we discovered that with continuous training, the males had a surge in serum testosterone. The females had a little bit of an increase, and it's not statistically significant, but it doesn't mean that it's not biologically significant. We've talked to several experts who deal in hormones, and they've said that that's actually quite a lot of estrogen, or a lot of testosterone to be found in a female. So it looks like the testosterone may be important. So we started to do a series of experiments where we tried to reduce testosterone. So the easiest way to do that is to castrate a male and then exercise them. So we had our males over here, our females here, and now we have our new group. And this is looking at fold, or the length of the axons relative to untrained. So again, one is going to be that untrained, untreated. And we can see that the males have that super nice increase with the continuous, the females don't respond. You castrate the males and they no longer have a response to that continuous exercise. So serum levels of testosterone, or gonadally derived testosterone, probably is important. Interestingly, it does not mean that that castrate animal is now a female. So if you interval train it, you do not get a response to interval training either. It's not that all of a sudden it becomes responsive to interval training where it wasn't before. So we do know that the serum testosterone is important. It was a little unclear what was happening with the females to get them trained. You can't just castrate a female to reduce their gonadal testosterone. So instead, we treated them with flutamide, which is an androgen receptor antagonist. So this is the fold change in axon peripheral length again, with the one bar being untreated. Here's our continuous treated male, our continuous treated female, interval, male and female. And then we have flutamide, which is our androgen receptor antagonist. And when you treat an animal with exercise with flutamide, you lose the efficacy or the enhancement of that exercise. So there's something about androgen receptor activation in males and females that are responsible for this exercise enhancement, which is super cool. So this kind of brought up the idea, could we use testosterone as a novel therapeutic for people who can't exercise? But I was still bothered by this result, because while androgen receptor signaling is important, why are the females only seeing a change during that interval training? And I thought there might be something else there. So we looked a little bit further, and I asked the question, could there be a role for estrogen? So with, if you know your biochemistry, some parts of testosterone or some of the testosterone molecules can go through aromatase and they can be aromatized to form estradiol. So could it be that these increases in serum testosterone that we're seeing with the males really be the aromatizable testosterone that then is getting converted to estradiol? And could estrogen have a role in this too? So we took these questions in my lab at the College of Charleston, and my students started asking them. So we started doing experiments with males and females where we treadmill trained the animals. And again, this is the number of labeled motor neurons, and the one line across here is going to be those untreated, unexercised animals. Again, treadmill training is going to increase this in males. It's going to continuous training is going to increase the number of labeled neurons. Just giving them estradiol by itself is a whopping increase in the number of labeled motor neurons. And this is true for both males over here and for females. So estradiol by itself is giving us the same kinds of increases that we are seeing with treadmill exercises. And I said, that's awesome. What happens if we block this estrogen receptor while we exercise? And we ended up with a situation. So ICI is an estrogen receptor antagonist. And if you treat the animal with it while you exercise in a male, it basically reduces the level of the number of motor neurons to about untreated animals. So you lose that enhancement of exercise. In females, it goes a little bit further. If you block estrogen receptors and exercise, not only do you lose the effect of exercise, but actually you go down below it. And if you give just the estrogen receptor antagonist by itself, you get a massive reduction in the number of motor neurons participating. So that suggests that there's something very important about estrogen receptor signaling, especially in females, for this participation in regeneration. So we thought that was pretty cool. We then said, well, what happens with sensory neurons? Because we can't just have motor neurons participate. What happens with sensory? So in this case, this is work done by Brenna Casey, Kira Bonfort, Catherine Epley, Josh Goodman, and Amanda Gosson. And I'm sorry, I should have told you, this work up here was done by Melina Acosta, Trisha Copley, and Jamie Harrell, and Allison Bruce. I like to talk about my students because they're the ones who are actually doing the work. So I want to acknowledge that this is their work. So again, here are the number of labeled neurons. We don't have that nice little white or that nice little bar, but you can see our untreated animals for females had about 200 sensory neurons labeled. Adding estradiol either systemically here or locally at the location at which the nerve cut happened increased the number of sensory neurons that were participating in regeneration by a lot, more than double. And then blocking estrogen signaling actually reduced that down to untreated levels. So we did not get any type of enhancement. And the same thing was found for males. So if you treat the males with estradiol, either systemically or locally at the site of the lesion, then you're going to get this large increase. And I want to make a point of why we actually did both systemic and local estradiol administration, because if you know any history of estradiol, or if you've taken a biochemistry class, you may know that if you try to treat a human with estradiol, it increases your risk of stroke by a lot. Early birth control pills had high estrogen levels and people had increased stroke risk. Trials that have used estrogen in the past have increased stroke risk. So we wanted to be able to come up with a treatment that was not going to just kill people when you're trying to fix a nerve injury. So a local administration could be an option. We were able to encapsulate it into a glue that was able to let it get into our axons, but we don't think it was going into systemic levels by monitoring both systemic serum levels of estradiol and also some physiological symptoms. So we think that we were able to maybe very locally treat these animals. So this is good news. So another potential mechanism has now come out. So it could be that we can use estradiol. So we now have this idea that exercise is somehow leading to estrogen, somehow leading to testosterone, and that maybe these could be some of these novel therapeutics. But it's kind of odd. When we're blocking estrogen receptors, testosterone signaling is still intact. When you're blocking androgen receptors, estrogen signaling is intact. So something else is part of the story, and we need to find that. because it's never an easy molecular pathway. You never have just one molecule that's doing it. These are complicated cascades. So we wanted to find out what's above this instead of going below. What's the marker above it? And I had a clue based on some research I did during my postdoc time that it could be a molecule of that neurotrophic factor, brain-derived neurotrophic factor, BDNF. And this is a molecule that we know that is upregulated very quickly after axonomy. So after nerve injury, it gets upregulated. And we know that BDNF and estradiol are very closely linked. When you overectomize an animal, its BDNF levels plummet. When you treat an animal again that's been overectomized with estrogen, its BDNF level goes up. So this could be some way, something that could be involved. So in this case, we have an animal, again, these yellow fluorescent proteins. They were aligned so that this white line is showing you the cut, and anything on this side of the line is elongated axons. Here's an untreated animal. It goes to about 1,000 microns, we saw before. The exercise animal goes to about 2,500 microns. This animal was just given brain-derived neurotrophic factor at the site of injury, and you can see it's elongated at least as long as the exercise, if not more. And then we created, I'm not going to talk about it a ton, but we created a novel BDNF knockout animal, which was super hard to do. And we were able to knock out BDNF and exercise the animal. And when you do that, you don't get any regeneration. This is really sad. So it shows that BDNF could be another novel therapeutic. Unfortunately, BDNF is huge and it doesn't cross the blood-brain barrier. And that's an important thing to be able to actually get it into a cell. So some colleagues at Emory came up with some BDNF mimetics. These are things that have similar characteristics to BDNF. They bind to the same receptor, but they are not the same. They came up with 7,8-DHF and deoxygenin as mimetics for BDNF, and they were able to show, and this time it actually looks yellow instead of green, that in these cases we have our 1,000 microns over here for our control animal, and with the 7,8-DHF and the deoxygenin, you can see you have a lot longer axons. So that these BDNF mimetics may be a novel therapeutic for engaging with axon elongation. I have not done the experiments yet to see if it has any effects on the sensory neurons or on the participation or on motor neuron participation. Those are some of the upcoming experiments they're going to be working on to learn about that. The interesting thing about these two drugs is that they are currently already being used by humans. You can find them at the GNC. People take them as muscle hardeners. So these things might be able to actually become, treatments a little bit faster than some of the other things that, I'm going to talk about. So now we're left with exercise, BDNF, estrogen, testosterone. Is there something that exercise is doing to get BDNF to upregulate? Because right now we know that those muscles that are being exercised during the exercise, they're not connected back to those nerves that are being injured or have been injured. Those have been cut, and we know that they've degenerated. So what is exercise actually doing? So to try to answer that question, we did some novel experiments using optogenetics. This is where we created a mouse that expresses channel rhodopsin, which is a channel that when activated by light opens up and allows sodium to come in and depolarize a neuron. So basically when you shine light on this animal, it's able to have those neurons have action potentials, or it can create activity without the animal having to do anything. So you can have an anesthetized animal, you can shine a light on its neurons, and all of a sudden the neurons start working. And if you know the pattern that the neurons should be working in, you could pulse it to get the action, to get the neurons to potentially look like they would while the animal is running. So we created this animal model, and then we took the nerve and we made a cuff to go around it. And inside the cuff, we lined it with lots and lots of little LEDs. We sewed the cuff around the nerve, then sewed the animal up, and we were able to turn on these, the LEDs, which activated the nervous system. We did this kind of like we did with treadmill exercise. We did it 5 days a week for 2 weeks. We then cut the nerve, applied our dye to retrogradely label these neurons, and we were able to count the number of neurons both in the spinal cord and the sensory neurons. And what we found is that the number of retrogradely motor neurons increased when the animals that had been treated optically. So it had that pattern of activation that was created by light, but it was giving some type of neural activity. So the neuronal activity increased the number of motor neurons participating in regeneration. And we looked at a longer time point. We did this at 4 weeks instead of the 3 weeks or 2 weeks we've been using earlier. And in this case, we could inject a dye into the muscle directly, into the gastrocnemius muscle, and then count the number of motor neurons whose axons actually made it all the way to gastrocnemius. So this was only neurons that had actually made it back to its target were going to be able to be labeled. And you can see that we get a huge increase in the number of neurons that have made it back to reinnervate that nerve, so compared to untreated. So this again becomes maybe a novel therapeutic. Could we use some type of stimulator? I'm not suggesting we're going to try to knock channel rhodopsin into humans and try to use them to treat that way, but maybe we could stimulate the nerve. Maybe we could stimulate the muscles and try to use that as a way to have a novel therapeutic. And it also helps us to fill in this gap. So somehow exercise is increasing neuronal activity that probably is increasing BDNF and estrogen and testosterone, and maybe that's feeding back. Maybe BDNF is up here, maybe BDNF is down here. We're not really quite sure how this fits together and what's really driving all of this. So this is kind of the model that we're left with and kind of some of the questions. So some new aspects of the research that we're trying to follow up is, what factors are exercise really doing to produce these changes in BDNF, testosterone, and estrogen? Could it be intermittent hypoxia? So when you're exercising really fast, could it be you're out of breath? Could it be a metabolic change? Could it be an immune system change? And then, when do these factors need to be administered to create the effects that we're seeing? So I've been talking about estrogen. We're giving that at the time that the injury happens. Most people don't have that luxury to go and say, oh, I just had an injury, let me immediately take, estrogen right now for it. Maybe in the future, but right now we don't have that. So when do the timings need to be done? So those are some of the questions that we have kind of coming up. So we kind of are left with this model. We have our treadmill exercise, continuous if you're male, interval in a female, trying to think about how we can retrain physical therapists to work together to try to treat patients. We think that this is probably increasing serum testosterone levels, tissue androgens. It's also increasing neuronal activity. Somehow that tissue androgen is probably going to get aromatized to increase estrogen. That's activating estrogen receptors. Our testosterone is activating androgen receptors. And then that together may be activating BDNF and increasing protein translation to enhance what we're finding. Interestingly enough, we're also found in our lab, and I didn't present this evidence today, but I did want to mention it, that in some animals we have them living in a cage where they have access to a running wheel, and it's just free access, and we have a counter on the running wheel just to count how long these animals were running. And sometimes the females at night will run miles. It's really amazing. When the males and females are treated with an estrogen receptor antagonist, they stop running. Totally. They just stop running. So there's some interaction even between this estrogen receptor and estrogen receptor activation and back this way to engagement and motivation to exercise. So there's a whole bunch of arrows in this, and we're still trying to fill in some of the blanks. Next time I talk to y'all, hopefully I'll get to fill in some more of these blanks, and we'll have even more words on this slide. We also are looking at the central nervous system. There's some research I have not talked about today. While it is really important for regeneration and participation in motor neurons and sensory neurons, we also know that these neurons are talking to each other in the central nervous system. So that motor neuron is getting information from the sensory neuron. That's why when you reach out to touch something and it's hot, your hand can come back. So that sensory neuron sending information to your motor neuron to say, hey, move away, even before it gets to your brain. One of the effects of injury is whenever that axon, or that's whenever the sciatic nerve is cut or the nerve is cut, the inputs from the synaptic or from the sensory neurons actually withdraw from that motor neuron. So this can cause functional problems. So even if you get the axons to regenerate, if it's not getting the right inputs, it all is kind of for nothing. So we have some wonderful students in the lab right now who are working to see what can we do? Are these types of treatments and therapies actually effective at that synaptic reorganization and for the synaptic plasticity? And luckily we've found so far, we have some preliminary results that one of my students presented yesterday at Abercans, suggesting that yes, estradiol and testosterone and BDNF actually are helpful at sustaining those synaptic contacts onto the injured motor neurons. So it's kind of good news that all these therapeutics are working for all the different metrics we have so far for this. So hopefully in the future, the question will be you go in to the surgeon and he's going to say, hey, do you want to see a physical therapist or a pharmacist? And maybe we can kind of work together, or they have those can both kind of work together to increase patient treatment. So with that, I want to introduce you to my lab. These are the current students I am so proud of. This is Shanice Wilson. She is a student at the college. She's a senior. Interested in pursuing PA school. This is Rochelle Peterson. She is also a senior biology major, interested in going into pediatric neurology. Of course, this is me. This is Vernon Kennedy Jr. He is a Goldwater scholar, woohoo, who presented at Abercams yesterday, and he is going on to get his MD PhD. He's applying to schools. This is Grace Bader. She's a sophomore in the lab. Kaylin Seidling, who graduated last year. And happy Veterans Day to Kenny Moreland, who is a non-traditional veteran who's now working in my lab. She is a freshman at the college and just starting out. And of course, I want to acknowledge my collaborators, our English, Jill Ward, and our funding. NIH, HHMI have both been generous with that, as well as my department. So with that, I'll ask if y'all have any questions.
Thank you. This was fascinating. Yes, students or colleagues.
Listen, I am so impressed. Not like that, but it's so relevant to real life. So I've had nerve damage, and so has I have athletes in my family. Oh, hold on. A poll came up.
I know. I launched the poll. I didn't want people to log off, and I might have lost them. But please do the poll. Sorry, students. Don't log off.
Oh, okay.
Please do the poll. I'm sorry, Dr. Morris.
Yeah, go ahead, Dr. Morris. You're muted.
What you're doing is great. And it was really relevant to my life, like learning about axon the regeneration. Like I had nerve injury to my pelvis, so I have had a pelvic break, and I tore some ligaments and stuff through childbirth.
Yep.
And so having to do the physical therapy and all of that throughout it, and it literally took a decade of me being in physical therapy still, like for it to get to a place where I can actually function. I was walking on a cane for a long time after it. And that child is 12 now. And so I am so happy about the work because the type of physical, like it took for me to change my regimen and say that all I can do is exercise every other day. I can't do this every day like you want me to do it every day. And I didn't know why, but it just made sense to me. I felt like I was just following my body, but you made it make sense scientifically. And so now I see why. And also the motivation to continue to keep going. That made a lot of sense. So I figured it was a hormonal connection, and you just made it plain. I'm so proud of that work. And I really hope that physical therapists listen to you and really take heed, because it's important. I mean, you're talking about people's quality of life. Now, we don't want anybody to stroke out, but we do need probably some supplemental hormonal therapy or something along with the treatment. And so I don't know. It just was so relevant. Thank you. Thank you for being here.
Oh, thank you for sharing your experience. I'm glad that you persisted and that you kept going on with physical therapy, because a lot of people just stop and they drop it off, and it's really important. And
I'm the stopper, but I'm going back. I'm going back. No, it was students. Before we jump into the old lady problems, no, I'm one of those. You hit it at the second diagram. I have L5 degeneration, S something down through there. And like literally I have physical therapy on Thursday to kind of figure out some stuff with me as well. But you have made mice look like they're the cutest creatures ever with that little hair bow.
Yes.
And I am not a fan.
Yes.
But
me either.
But I will say, I feel like, I mean, it's on the brink, and I hope you're going high impact for that work, for nature or science or something. Just go hard with it for that work. Yes, please, because
it needs to be public knowledge.
Yeah,
Like for
and
real.
the thing is, we've seen some differences in cartilage with knee problems, the differences with male and females. I don't know if they've looked at hormonal, but when you first said it, I was like, what's the hormone? That was my question, and you followed it. There's so much I have to say, but I'm going to kind of keep it to a minimum. For the later part of the work, I have two questions. Guess who has a hypoxic chamber? For one of those last research questions, me. So we have a huge biospheric hypoxic chamber where we can do hypoxic work in our chamber, and also do incubation studies as well. So I'm happy to offer my collaborative efforts to you for that. But the other question I have, was two, but I'll save them for later, is do you think having a synthetic nerve, if I don't know if anybody's ever tried to make one, a biosynthetic nerve, do you think that would show promise, or does it just depend on the makeup? Would it show more promise than cadavers, or is it still just something that hasn't been touched upon yet?
That's actually been worked out pretty well. Ravi Valamkanda, who used to be at Georgia Tech, and I believe he is at Rutgers right now. He does amazing work, and that was some of what he was looking at, is what biomaterials are important to use. How do you, so if you have the biomaterial, you need to seed it with something, because a lot of the growth factors are being released by these reactive Schwann cells. And when the axons are growing back and they make contact with the reactive Schwann cells, those reactive Schwann cells transform and become myelinating Schwann cells again. So we end up in a situation normally where the Schwann cells are basically pulling that axon down by having a gradient of neurotransmitter factor. So one of the questions and one of the problems has been, how do you seed a biomaterial so that it has the appropriate growth factors on at one time, and then it turns off when contact is made? So they've done some really interesting things with looking at resistance, where something leaches out a material, and then as soon as the axon grows through, it changes the resistance in that area so that it shuts off for it to leak out, to kind of draw it through. You still have misdirection. You still have issues with things not actually growing where they need to go, kind of going out to getting neuromas. There's going to be a lot of challenges with going that way, but it is a promising area of research.
Yeah, so I'm curious, because even we have the capability in my lab where we were the first to publish on, we electrospin fibers, and we can essentially do that with kind of guiding them. So we can essentially make a biomaterial that's shaped like a nerve, right? And align fibers in that capacity, and then have them porous in a sense. I don't even know if anybody's done that, but I'm curious to know if we kind of literally try and mimic it. We could probably 3D print it too, so that they can align properly, if that would help with some of the, and maybe take an actual, we can talk later, because y'all know, the students know I can just go down this path and I won't come back. It'll be the point of no return. But anyway,
I'm here for it, girl. I'm here for it.
It will be curious to see if we can fabricate, because that's what so we can fabricate it in a sense where it might help with the alignment and essentially the diffusion of those things. I don't know, but I may give that to a new student that comes in the lab that may be interested in neuroscience, to kind of look at some of that stuff. But we do have a capability of kind of doing that. And it might be something to look at in addition to the hypoxia. And I bought a new tool that we can do real-time cell imaging of labeling or whatever we want to do in our materials under hypoxia as well. So everything that we kind of incubate under hypoxia, we can do. So it might be, we'll keep talking, because I'll get on your nerves. And we also would be able to image those. So spatially, we can see where everything is and kind of figure it out, even biochemically for you with some of your BDNF stuff.
Yeah,
we can actually see where that's going. But I'm going to stop, because I can keep
students
going.
on the call, what you're hearing is science in action. This is how science is done. It's not, it's done by these kinds of conversations. And there's also potentially an opportunity. So what y'all are hearing is that if Dr. Mendenhall and I can work something out, there might be an opportunity for y'all to come to Charleston, South Carolina, to do some of this research, to learn how to do.
And I've never been.
And Charleston is an amazing place. If y'all have never come, we have great beaches and are, a really wonderful, welcoming environment.
And food, I'm sure too. So no, that would be fantastic. And she's a great teacher, because if you didn't learn anything, then I mean, I learned a lot. So I think it would be, and the beauty
No,
of
I
it
learned
is
a whole lot.
students to also take away like how she even took biology and she combined a lot of other different things into her talk. So it was biology, anatomy, physiology, and kind of pulling all of these things together. It wasn't just centered. And that's the thing we have to remember as you matriculate. When you take a bunch of different hodgepodge classes, there is synergy that comes between you actually pulling all of the knowledge you have. I'm a chemist by training, and you see I just kind of went into, I can figure out a way to work with her. So those are the places where we push you and we encourage you to continue to, use your research capacities and learning how to read literature and figure out how to, contribute in other spaces. Do we have any questions from the students? Because Dr. Morris and I kind of hijacked it with our excitement. But students, please raise your hand or speak up, unmute yourself.
As y'all are thinking about your questions, I want to echo what Dr. Mendenhall just said. I think it is really important to think about the intersectionality of your knowledge and to really, embrace that and embrace the breadth of what you can experience at a liberal arts college. So I was trained at a liberal arts college. I went to Davidson undergrad. I have really wanted to I love working at liberal arts colleges because you can take so many different things. I teach in a psychology department, but you can see my research is very chemistry, biochemistry, heavy. And a lot of my research, what I did earlier was very heavy into physics. So you can take these different things and work them together. And that intersectionality is where the answers to our big questions really tend to lie. So it's going to be important to do that. I also want to encourage y'all, if y'all are doing research, keep persisting. You're going to see that there are going to be some challenges along the way. There are going to be things that you think are going to go one way and are going to turn out a different way. And you may end up with you may hit some roadblocks and some people who are less supportive. Dr. Mendenhall and I have talked quite a bit about different people who have maybe gotten in our way and told us that we needed to be doing something else. And it's important to keep persisting. It's important to keep working and to keep challenging and to keep pushing, because what you're doing is important, and what you're, where you're going to be able to go and what kind of discoveries you're going to make are going to be really important. It's going to be important for you. It's going to be important for other people, and your voice is worth contributing. So I want to make sure that you understand that there are going to be challenges if you want to become a scientist. It's not an easy road, but it's one that's doable and it's manageable, and it's important to have good colleagues and good mentors. So it's been invaluable to me. I mentioned Dr. Chena Chang earlier and Dr. Jackson, and they were both instrumental in teaching me really important things along the way. And Dr. Val Hachtel, that some of y'all might know in the biology department, and Dr. Mendenhall too, have been really helpful to teaching me how to teach, how to be a mentor, how to do research. So it's going to be important for y'all to reach out, to have advisors, to have mentors, and have sponsors, people who are going to really push you along. If anybody wants to get in touch with me, I'm going to put, I'll be happy to put my, I was going to look for a chat bar. Yeah, there's a chat bar. So I'm going to put my email address in the chat bar, and y'all are very welcome to